There are a wide range of in vitro assays that use LC-MS for detection, including clearance, permeability, plasma protein binding, and enzyme induction and inhibition. In this talk, we'll look at what's involved in the analysis of such assays. Generally, sample preparation for in vitro assays follows a similar pattern once the incubation or reaction is complete. An organic solvent, typically acetonitrile containing an internal standard, is added to precipitate any protein. Water is then added to the samples and the samples centrifuged before analysis. In this talk, we'll look at why we add both an internal standard and water. From an analysis standpoint, we'll look at the instrumental requirements in terms of the limit of detection, the linear response range, and then we'll indicate some of the problems that can arise. For more details on how the various in vitro incubations are performed, I've copied a link to the videos on how to set up both clearance and permeability at the bottom of the slide. So why do we add an internal standard in vitro assays? Well, if we look at analytical chemistry, the internal standard is added to the sample prior to any sample pre-treatment or preparation and is used to correct for any losses that occur during those procedures or indeed during the analysis. The internal standard is chosen to have physicochemical properties as close as possible to the analyte, often a stable isotope labeled compound or a structural analogue. In bioanalysis, the internal standard is added after the incubation. In effect, it's a test of the analysis only. Most in vitro assays are performed of tens of compounds simultaneously, and a specific internal standard for each analyte is impractical, so we use a generic compound which is stable analytically. So what do we look for in terms of the internal standard response? As the internal standard concentration is identical in each sample, we'd expect identical peak areas. The example in this table is from a clearance assay showing 6 time points. The average peak area for the internal standard is shown at the bottom, and all the samples are within plus or minus 3%. Generally, a tolerance of plus or minus 15% is set for each assay. If a sample is out with the 15% range, they should be discarded. Occasionally, there's a gradual decline in internal standard response, and this can indicate problems with the mass spectrometer. So why do we add water? It's basically to ensure a good chromatographic peak shape. If the sample solvent has a higher elution strength than the starting composition of the gradient, it can lead to problems with fronting peaks. An example of such a peak is shown in chromatogram B. Ideally, the sample would be dissolved in a similar elution strength solvent to the starting composition of the gradient. But to do so would require samples to be dried and reconstituted, not only time-consuming, but would also certainly lead to solubility problems. As a compromise, water is added to give a solution which is 50 to 60% aqueous. We can look at the effects of different sample solvents using caffeine as an example. Here, run on a typical fast LC gradient used for clearance samples. The LC gradient has a starting composition of 95% aqueous, which is held for 0.3 of a minute before a linear gradient to 5% aqueous, held there for a further half a minute before returning to the starting conditions. If the sample solution consists of 67% aqueous, 33% acetonitrile, the peak shape is good as shown in peak A. Increasing the percentage of organic to 66% gives the peak fronting shown in peak B. And if we inject caffeine in 100% acetonitrile, it's eluted continuously, as you could see from the raised baseline on chromatogram C. Almost all in vitro work using mass spec is run on either triple quadrupoles or high resolution mass specs, generating either MRMs or single ion monitoring. Chromatograms for the analyte and internal standard are smoothed and integrated, and the peak areas tabulated and recorded. The area for the analyte and the internal standard are then used to calculate a response, which is simply the area of the analyte over the internal standard. It is important to set some form of minimum detection level for data. This can be done following data processing when a manual check of the data is performed and estimating roughly a signal-to-noise ratio based on the background noise. An alternative to set a minimum peak area and reject any data below this level. This is simpler to implement but can lead to perfectly acceptable data being discarded. The example here shows a relatively weak peak indicated by the asterisk, which is just above the signal-to-noise limit of 5 to 10, which is often set. In this example, the signal-to-noise can be done by simply eyeballing the data or by magnifying an area of the baseline close to the peak. Here we've magnified the baseline by 10, and it shows that the peak has roughly a signal-to-noise of around 15 to 20 to 1. As noted from the previous slide, it's important to check detection levels for compounds in terms of signal-to-noise, but the question of detection levels can also be looked at in terms of assay requirements. For the example here, we'll compare a microsomal clearance assay with a permeability assay using MDCK cells. For clearance, we are trying to find the rate of turnover of a compound, and for highly cleared compounds, we generally need to see down to approximately 1% of the initial concentration in order to generate sufficient data points to give an intrinsic clearance value. The example here shows verapamil and gives the peak area of verapamil versus time. The initial peak area is around 50,000, the final peak area around 1,000, so approximately 2% of the initial starting concentration. For permeability, we're looking at diffusion of compound through a membrane, and for comparison looked at two compounds, atenolol and bupropion. Atenolol is poorly permeable, and as you can see, the level in the acceptor well of around 1,000 is much lower than that in the donor well, which is around 1.5 million. For atenolol, we now need to see a much greater linear range, with the acceptor being around 0.05% of the original concentration in the donor well. Both assays use different incubation concentrations. Permeability assays are often performed at 10 micromolar, whereas clearance assays are performed at a much lower concentration in order to prevent saturation of the various metabolizing enzymes. Typically, clearance is performed at half a micromolar starting concentration. However, following dilution with the internal standard and organic, and then water, the initial concentration to be analyzed is now much lower, in this case 0.063 micromolar. As mentioned previously, we need to see down to 1% of this, so the instrument should be capable of detecting down to 0.63 nanomolar. For a compound with a molecular weight of 400, I've put the equivalent concentrations in terms of nanograms per mil, so we would need to look over a range from 250 picograms per mil to 25 nanograms per mil. For permeability, the incubation concentration is 10 micromolar, and the concentration seen for analysis is now 1.25 micromolar. For poorly permeable compounds, we may need to see down to 0.01% of the initial concentration, 0.125 nanomolar. Again, I've put the equivalent nanogram per mil concentrations for a compound of a molecular weight of 400. So that would be 50 picograms per mil to 500 nanograms per mil. For electrospray, the linear range of compounds is often limited, 3 orders of magnitude or less. The response of a compound over the range required for a permeability assay will often approximate to a non-linear quadratic response rather than linear. Even where compounds display linear behavior, the regression line in electrospray has to be weighted as 1 over x or 1 over x squared. What are the effects of nonlinearity for in vitro assays? Most in vitro work is done by comparing response between time points. No calibration line is used, and the response is assumed to be linear and correspond to concentration. The effect of nonlinearity can be seen in the diagram. If we have two samples, P1 and P2, of different concentrations, and the response of the detector is linear, as shown in the blue, then the ratio of the intensity or the response is equivalent to the ratio of the concentrations. However, if the compound displays a more quadratic response, as shown in the red, the peak intensities for P1 and P2 clearly do not match their concentrations. Without generating a calibration line, we don't know if our compound displays a linear or nonlinear response. And if it is nonlinear, what is the degree of that nonlinearity? The response of compound A1, shown in green, gives a close approximation to a linear response, whereas A2 clearly does not. Because of this, we shouldn't place too much emphasis on absolute values from in vitro assays, such as CLINT for clearance or PAP for permeability. Instead, it's preferable to categorize compounds in terms of low, medium, high clearance or permeability. So what can go wrong? Luckily, the talk is only 15 minutes long, so we can't detail all possible issues, but we can look at one chromatographic and one mass spec problem. Polar compounds can be difficult to retain, and the upper chromatogram shows a peak which starts eluting around 0.55 minutes. The void time for an unretained compound is approximately 0.25 of a minute in this system. It's worth noting that the void time is different for each LC system. The gradient is the same as used for the clearance samples we looked at earlier, and there is a 0.3 minute delay between injection and the gradient starting. The compound is therefore barely retained, as the relatively broad peak shape indicates. There is a greater likelihood of peak suppression, as the peak will be eluting with other unretained components such as salts, solvents, and unprecipitated proteins. By changing eluents to basic condition, the peak is retained and gives a much narrower peak shape. If a change of pH does not work, different columns can be used. Most work is done using a C18 column, so other column chemistries such as PFP or even changing to a Hillick system may be appropriate. In terms of mass spectrometry, problems can arise with compounds being poorly ionized, and therefore it's not possible to achieve the detection levels required for the various assays. The use of an alternative ionization mode, such as atmospheric pressure chemical ionization, APCI, can often improve the sensitivity for such compounds. High background noise is another problem that can occur. Although the use of MRM and high-resolution single-ion monitoring can greatly reduce background noise, occasionally this is not the case. In this example, a compound gives a high background noise on a particular MRM channel. The signal to noise for the peak is only around 5 to 1, even though the peak height exceeds 120,000 counts. By using a different MRM transition, the peak height is reduced by half, but the signal to noise now exceeds 100. If this problem occurred on a high-resolution instrument, increasing the resolution may help, or alternatively trying to change the chromatography in order to separate the source of the noise from the analyte may do the trick.